Hello, welcome back. This is Professor Young, and this is part 2 of our look at the cell membrane. And this is a part 2 of 2, where in this session we will talk about channel proteins and a little bit more about the overall structure of everything. And so that is kind of where we are heading with this session here, looking at channel proteins. Looking a little bit at active transport, not too much, but a little bit. And again, taking that closer look at exactly what makes up a cell structure as far as giving it support and shape. And prior to this, in our first session, if you recall, we were introduced to the overall shape of the cell membrane. We understand that it's this phospholipid bilayer that has a hydrophilic and a hydrophobic end to it. We understand that built within that, that there are dispersed throughout these integral proteins and these peripheral proteins, and that together the phospholipid bilayer, the integral proteins, as well as the peripheral proteins, all come together to create this concept of a fluid mosaic model, and that specifically looking at those integral proteins, where we left off was talking about this concept of a carrier protein and some of the characteristics of those carrier proteins. And so with that said, just as a little bit of a recap, let's go ahead and continue on our way looking at channel proteins. Channel proteins differ from carrier proteins in a couple different ways. First of all, channel proteins are prolific at the rate at which they can transfer typically ions from one side of the cell membrane to the other side of the cell membrane. We're talking about tens of millions of ions per second, whereas our carrier proteins were doing anywhere from about 1,000 to 1 million. Now our channel proteins are doing tens of millions. 20, 30, 40 million ions per second being able to cross. And part of that comes from the fact that these channel proteins, think of a channel, a physical channel that would go ahead and go through a waterway. Right? They're constantly filled with water. All right? A channel, a physical channel, is usually filled with water that a ship can then go ahead and move across. Well, channel proteins are kind of the same way. They're water-filled channels that connect the exterior and the interior sides of a cell membrane. Now, these channels can be specific. They can be very specific, or they can be based, let me rephrase that. They are very specific. And the specificity is coming from typically either a size or a charge. So if the channel is a small channel, it's going to reject the larger ions. And if the channel is positively charged, it's going to repel negatively charged. And if it's negatively charged channel, it's going to repel or it's going to attract positively charged ions. And so there is definitely some structure for that. There is definitely some structure there, and organization and can't think of the word I'm looking for, or control mechanisms that are in place that are going to dictate what exactly can get across this channel protein, and that's needed. It's needed because of the simple fact that there's nothing else regulating what's going to be able to flow through that channel protein. And so having that specificity based on size or charge is going to go ahead and help regulate to some extent what exactly is crossing and what exactly is not crossing. Now, as I've already mentioned, that selectivity or the specificity can be based on the diameter or it can be based on the electrical charge. And so this is kind of one of those things that I've already touched on that previous slide. And with that, one of the things that I do want to kind of stress here is that there are two types of channels. There are open channels and there are gated channels. Open channels are just that. They're open. They're open 24/7 or practically 24/7. They're not overly picky about what flows through them. On the other hand, we've got these gated channels. And gated channels do tend to have a higher instance of specificity built into them. In fact, they spend most of their time closed instead of being open. Now, at the onset of this, I did say that channels are almost continuously open, as opposed to their counterparts, that being the carriers. Right? And here's the thing. Gated channels might appear to go against that notion that channel proteins are almost always open for business. But if you think about this, right, the main difference between an open and a gated channel is that gated are a little bit more regulatory. They're a little bit more picky about what's coming through. What classifies or what really makes that gated channel a channel and not a carrier protein is the way it functions. That gated channel, by far, is regulated through water passing through it. And that's the key determination. If you remember and look back to what a carrier protein is, that molecule or that ion has to fit into a receptor. And once it binds into that receptor, it causes a change within the shape of the channel that closes the one end and opens the other end. That's what makes that a carrier protein. What makes this a gated channel is the fact that even though you have an extremely high amount of specificity and regulatory control going on here, what we do have here is when that channel is open, the floodgates are open. And you again have 20, 30, 40, 50 million ions traveling through that channel at the same time. And so that's a big difference here. All right, that's a big difference here. And sometimes people question that and they say, well, if channels aren't as regulatory, what's up with these gated channels? And that's what's up with them. Yes, they're regulatory, but when they're open, the floodgates are open and everything's moving through. Now, there are three types of gated channels. These channels can be controlled by a chemical trigger, right? A chemical change, typically the binding of a protein or an enzyme that will open up that channel. It could be a voltage trigger. In other words, a change from a positive to a negative charge, overall charge, or net charge of that channel. Or it could be a mechanical trigger. Right? Something physically is going to go ahead and open that. It's not a chemical change. It's not a voltage change. It's a biological aspect that's going to go ahead and cause a change in this overall structure. And so we see a little bit more complexity and we see a little bit more breadth, if you would, or differences within our channels than what we did with our carrier proteins. A carrier protein is a carrier protein, but we do have these different types of channels that we're dealing with. What I'd like you to do for at least a couple minutes, and I'm just going to kind of sit here and let you wander about this, see if you can identify or answer these four questions. Can you identify the types of membrane proteins represented by peripheral proteins? Can you identify membrane proteins that are considered integral proteins? Can you tell the difference between osmosis and facilitated diffusion? And what are some major differences between channels and carriers? I'm going to stop for, 15, 20 seconds. But remember, you can pause this to be able to answer these questions a little bit more actively. All right, so hopefully you've had a little bit of success in being able to answer these 4 questions and really think about what these differences are. Specifically, the last 2 questions. What's the major differences between osmosis and facilitated diffusion? And the big question here is the differences between channel and carrier proteins. A couple minutes or a couple seconds at least on active transport. I would be a horrible A&P professor if I did not at least talk about active transport for a couple minutes. The bottom line here is, and we've talked about this before, it comes down to that guy right there. It is all about him. It's all about ATP. All right. ATP is the energy that is needed in order to have active transport happen. And when we talk about active transport, we're talking about the movement of molecules and or ions with the physical usage of a pump. Right? What we often call a molecular pump. And these molecular pumps are designed to pump ions against their concentration gradients. Right? These molecular pumps are designed to pump ions against their concentration gradients. When we talk about a concentration gradient, we're talking about over a span of whatever distance we're dealing with, typically maybe over the span of a cell membrane. The distribution of ions are not equalized. You may have more of an ion on the outside. You may have fewer of that ion on the inside. The movement of those ions under normal pretenses is always typically going to be from an area of higher concentration to an area of lower concentration. Well, sometimes what ends up happening here is we need to go from an area of low concentration to an area of high concentration. We need to go against that natural order of ions, and so that is where a pump would come into place. Or sometimes the ion is just so big by itself that it's not going to fit through a channel protein. Carrier proteins aren't going to work. And so we've got to go ahead and push. We need a little bit of oomph behind this to go ahead and have everything work and go and function as it's supposed to. And so that's where ATP comes in. ATP is going to supply the energy for this channel, for this pump, to move the ions in the direction that it needs it to go. And again, as we already know, when we break ATP down, when we break that third bond, between the second and the third phosphate group. Once we release that third phosphate group, we create ADP, adenosine diphosphate. Now, taking this a step further, when we talk about pumps and the way that we name them, their name tells us everything. Right? The name tells us everything. And so as an example, We've got calcium ATPase. All right. ACE here, ASE, you should recognize is an enzyme. Most of our pumps are going to be some kind of, is going to function off of an enzyme, but it's protein based. And this is a pump or an enzyme that is going to require energy in the form of ATP, which is going to move calcium ions across the membrane. And so calcium ATPase is going to be a protein that is going to use, or it's going to be an enzyme that is going to rely on ATP to move calcium across the membrane. Now, active transport tends to not want to waste the energy. It wants to conserve as much of that energy or get the most potential out of that energy. And so to do that, we have two types of transportation mechanisms at work here. The first one here is co-transport. And with a co-transport, that pump is actually not only just moving one ion, it's moving two ions. And it's moving both of those ions in the same direction. We typically see this happen quite often within the kidneys. And when we get to the kidneys, that'll make more sense. That's AMP2. The other variety of transport pump that we have here is countertransport. And countertransport here, as you might suggest or think about, is going to take two ions and it's going to pump them in opposite or opposing directions. And the good old standard example of that is sodium-potassium ATPase. This is a pump that's going to utilize ATP, and it's actually going to move sodium and potassium across the membrane. It's going to kick 3 sodiums out of the cell, and it's going to bring 2 potassiums into the cell. And when we do that, when we kick 3 sodiums out and bring 2 potassiums in, what that triggers is an action potential, which then will allow the muscles to contract. All right, last little bit. This is a short WIMBA, especially compared to what you're used to. As I said in lecture, don't worry about that. I should have taken that out of here, and I didn't. Don't worry about that. All right. What we want to talk about here for a few seconds, though, is the cytoskeleton. What exactly is going to give the cell its shape and support? What's going to keep that cell from collapsing in on itself? And this is critical because even though we don't tend to think of that something that small needing support, something as small as a cell, it definitely does. And so the purpose of that cytoskeleton is to give structural support to the cell found within the cytoplasm. And there are basically 3 components to the cytoskeleton. We have microfilaments, microtubules, and intermediate filaments. Of these, the ones that are of most importance to us are the intermediate filaments. And they're going to become more critical to us in the short term because of our look at tissues. Microfilaments are going to be another key player as we get a little bit further down the road during this semester. Don't forget about those microfilaments, and we're going to talk about them in a lot more detail. Again, once we get to the muscular system. But for the immediate future, the intermediate filaments are going to be a key group for us. So when we talk about microfilaments and microtubules, micro because they're both very small, right? But microfilaments are by far the thinnest diameter-wise of our cytoskeleton, and microtubules have the widest or the largest diameter out of all the components of the cytoskeleton. And so microfilaments, smallest diameter, microtubules, largest diameter. And then there comes probably the more important aspect here, and that is the intermediate filaments. There are 5 basic intermediate filaments found within cells. Right now, each cell is going to have one of the 5 types, and these 5 types are specific to the 5 types of tissues that we have within the body. All right, and so these 5 intermediate filaments right here are all linked to a specific type of tissue. So keratin is linked to epithelial tissue. And so tissue or cell types that are epithelial, whether they're squamous, whether they are cuboidal, whether they are transitional, whether they are columnar. Epithelial cells have keratin as their intermediate filament, giving it support and structure. Vimentin is the next one. And vimentin is the intermediate filament for connective tissues. For connective tissues. And so your loose areolar, your dense regular, your dense irregular, your adipose tissue, your bone, your cartilage, your blood, and there's a few in there that I know I'm forgetting. Those cell types that are deemed connective tissues, their intermediate filament within their cytoplasm is vimentin. Muscular tissue relies on Desmin as its intermediate filament. Nervous tissue is going to rely on neurofilaments. And I know, as I have mentioned on a couple occasions, there's this goofy oddball tissue type called glial, and it relies on GFAP. And we will define what GFAP is a little bit later in the semester. But for right now, GFAP, for this purpose, is the intermediate filament that is associated with glial tissue. And again, one intermediate filament type for each type of tissue that we have. And so that brings us to an end for Chapter 3, or our look at the cell membrane. Where we are heading next is going to be diving into a little bit about DNA, a little bit about how it replicates itself, and more importantly, how we go from DNA to proteins. And so you can be looking forward to that WIMBA, as it's probably one of my more favorite topics in A&P1, because there's a lot going on there, and it is the basis for everything that we are, that we were, and that we will be. And so with that little teaser, I will leave you to study your cell membrane structure, function, purpose. And as always, if you have any questions, please do not hesitate. Give me a shout, give me an email, jot it down, ask me during the next lecture or lab. Always happy to answer those questions.